Okay, these are my glucose molecules. This is the alpha glucose, and this is the beta glucose. Sorry, it's kind of cramped here. The main primary difference that identifies is the placement of this hydroxyl group off of the first carbon atom. For alpha glucose, it is found below, which makes it less stable and more reactive, and is easily digestible, which causes things like the formation of starch. And then the hydroxyl group above the first carbon atom in beta glucose is found more common in nature, things like cellulose, plant walls, things like that. For the sake of this first model, I'm going to switch these groups because this is what allows for the... Okay, sorry, I have paused the video, but I switched these, so now these are both alpha, oops, is this right? Yeah, alpha glucose molecules. And, sorry, I had to check the rubric really quickly. The first molecule I'm going to make is going to be maltose, which is the two bonded glucose molecules. So what's going to happen here is these two hydroxyl groups are going to, they combine, except they don't combine. What happens, the molecule is, it removes a water molecule, which would be these two hydrogens, H2O, two hydrogens and a water, and then they combine on, oops, sorry, I had to rearrange some things, but they combine along this oxygen molecule, which was attached to this glucose. And this formation, with the removal of the water atom through hydrolysis, or it'd be dehydration synthesis, is what forms the maltose molecule as the two bonded glucose molecules. Okay, I did some moving around. The next reaction I'm going to be forming is dehydration synthesis once again with my fructose model and a glucose model. This is essentially the same process. It removes a water molecule, which will once again be this group here, this hydroxyl group, and this hydrogen atom. And then they will bond along this. And they will bond along this oxygen atom, which with the removal of the water is dehydration synthesis. And this forms our sucrose molecule. Okay, I rearranged my molecules again. My two simple sugars, glucose molecules, and my fructose molecule. Now, these individually make up monosaccharides, which is a simple sugar, just like a singular glucose molecule. When we bonded the two glucose molecules into maltose, it created a disaccharide through dehydration synthesis with the removal of a water molecule. And in comparison, things such as starch is a extremely long chain of these connected monosaccharides, the simple sugars, and they form a starch, which is, well, a very basic example, they would form a starch, something such as a starch, which is a polysaccharide. Now, all of these molecules I have made so far are carbohydrates, which are primarily used for, as far as in humans, immediate or non-immediate energy storage. So, and the reason the structure of this molecule is so important for this is all of these bonds strand out, which allows for which makes it very easy for these bonds to break, and that will release energy and allows the body to break down, which is more specific in this alpha glucose molecule because the placement of this hydroxyl group. But in, let's say this was a beta hydroxy group, beta glucose molecule was up here, and this hydroxy group was placed here, and the hydrogen was down here. It is still used for energy, just in a different form. To continue what I was saying before, these molecules, the sugar molecule, also is important because it will enter the ATP cycle in glycolysis, which will break it down into a pyruvate, which releases ATP, or it causes one of it releases energy due to one of bonds being broken. All right, here we have my lipids. We have the unsaturated fat, the saturated fats, and the glycerol. The glycerol will be used for my models later on, forming a diglyceride and a monoglyceride, and explaining a triglyceride. So the main difference between the saturated and unsaturated fats is found with the actual structure. In unsaturated fats, there is this double bond between the two carbons, which fills out the carbon bonds. And comparison to the saturated fats, each carbon bond is fully paired with these hydrogen bonds. It is matched to as many as it possibly can. Compared to this, where it is double bonded, a carbon is double bonded to another carbon. Here you'll notice there are no double bonds within these carbons. The only double bonds you will find are to these oxygen atoms. Okay, to start off, I did some rearranging so I could do this properly. I will be forming a monoglyceride. And so to form monoglyceride, for the glyceride we have these 3 hydroxyl groups. And to form the monoglyceride, we are going to take away a water molecule, so dehydration synthesis basically. So we take this hydrogen off the hydroxyl group, and then we take the entire hydroxyl group off here. And then along here, I'll shift this way just a little bit. This oxygen then bonds to this carbon atom as such. And this is what forms the monoglyceride, which is removing a water molecule. Now the formation of the diglyceride is a very similar process, almost exactly the same actually. We are removing the second hydroxyl group off of the glycerol. Sorry, this is hard to do with one hand. And then we are removing the hydrogen off of the hydroxyl group of the saturated fat. And then I'm just going to shift this over and, bond. And then the oxygen, once again, bonds with that carbon atom. And then this forms your diglyceride. And to form the triglyceride, you would essentially take another one of these saturated fats and attach it to this bottom group, which makes the most common, just like lipid fat, is a triglyceride. Okay, so what I have here is a phospholipid. And the primary difference between the phospholipid and a diglyceride, for example, is here on the last group of the glycerol, instead of having another fatty acid chain, we have a phosphate group, which is the oxygen, the hydroxyl double bonded to an oxygen with another oxygen. And then a characteristic of the phospholipids is that it typically, the most common found is a saturated fat and an unsaturated fat, which is, once again, the unsaturated fatty acid has the double bond with the carbons, and the saturated fat has all of the carbons bonded to as many hydrogens as possible. Now, within this phospholipid, there are 2 sections, a hydrophobic and a hydrophilic section. The phosphate group is hydrophilic, and that is usually demonstrated by the end sticking out the other direction, which is attracted to the water molecules, and it kind of, it just attaches to the water, while this end is away. These are the fats. These are hydrophobic. These do not like the water molecules, and they will repel water, basically. The phospholipids' polar, nonpolar structure allows it to be a semi-permeable surface. So, for example, in a cell, the phospholipid bilayer on the outside of the cell is a semi-permeable object that allows things within the cell to leave and re-enter the cell. Not whatever wants to leave and re-enter the cell, but for specific purposes. And yeah. Okay, so here I have my 3 models. I have my 2 alanine models, which I will use for my later formation of a dipeptide. And then here I have my generalized amino acid, which has the NH2, the CH with the R group, which I'll explain in a moment, a CO, and a hydroxyl group. And then, so the R group is basically just what differentiates each amino acid into a specific function. It determines things like polarity, whether it's hydrophobic, hydrophilic, etc. Okay. Now, here we have the 2 alanine groups. And to form a dipeptide, it is, in this case, and in most cases, breaking off hydrogen through a water molecule through dehydration synthesis, and then bonding through a single peptide bond. So for this scenario, we have this hydroxyl group, which will unbond from the alanine. And then we have this single hydrogen group, which will unbond from the nitrogen. And then these will bond, forming that single peptide bond I was just talking about. Whoops. So that would bond there, and then we need to reconnect these as such. And then this would form the dipeptide. In this case, it is 2 alanine groups bonded together. Now, the hydrolysis of this molecule, of this dipeptide, is going to be the reverse. It is going to be splitting it back into its single amino acid form, in this case the 2 alanine groups, by adding back the water molecule that we just removed. So what will happen is here, where we just wrote this peptide bond will break. Oops. Sorry. And through this bond, we are going to add back on the hydroxyl group, the hydroxyl group that was broken off of the first atom. I need this one. Sorry, it's sticking to my hands here. I'm going to add back the hydroxyl group, the OH. And we are going to return the hydrogen atom to the other one, the other alanine group. And this would be the dehydration synthesis of the 2 alanines that formed the dipeptide, because we added the hydroxyl group and a hydrogen atom back that were removed originally to form the dipeptide, and the hydrolysis basically just broke that bond and added the water molecule, the hydroxyl group, to 1 and 1 hydrogen atom from that water molecule. Okay, so here we have my 2 models of deoxyribose and ribose. Sorry. Deoxyribose is used in DNA. Here we have the main identifier between the difference of the deoxyribose from the ribose is on the C2. You can see I vaguely numbered these. I don't have the prime symbol, but those are going to run off the side. But this would be carbon 2 prime. This one has a hydrogen group, just hydrogen, compared to this C2 has an hydroxyl group. The deoxyribose does not contain the hydroxyl group, while the ribose does contain that hydroxyl group. And up here we have my just generalized phosphate group, nitrogen group. Over here we have it as well, phosphate, nitrogen, and these are unrelated parts. So yeah, these are both monomers or single building block units, which is the deoxyribose and just ribose. Okay, here you can see I have remodeled, I changed my ribose group into a deoxyribose group, so I have two DNA monomers which I connected through the C5 and C3, the carbon group 3 and carbon group 5. The carbon group 3 attaches to the hydroxyl of the carbon group 5. So they just share this hydroxyl. And this shape, this general shape, if you continue to connect all the monomers, it will turn into a double helix, which is responsible for storing DNA. And this double helix or base pairing creates an anti-parallel with the other side of the double helix, which is essential for coding for amino acids in the human body, which everyone's are different, which once again with DNA or deoxyribonucleic acid, which contains the ribonucleic acid or deoxyribose, these base pairings that are caused by the double helix in the anti-parallel pairings of the amino acid, with code for the amino acids, give everyone their unique characteristics and traits.